anniversary that I never thought I would see. We're at our 1-year anniversary of the COVID-19 global pandemic. And to mark that, Dr. Collins has put in a really nice editorial in Science. It just came out, at least to my desk this morning. It's worth reading. There's a lot to be proud of in what we've done in vaccine and vaccine development. And there's still a lot more to go. And so if you have the time, it's worth reading that editorial. Well, I want to thank you for coming today. If you want to go to the next slide, Erin. The seminar series is sponsored by the Office of Data Science Strategy. There's a plug for our office and some of the ways to connect with us. But moving on right along to the next slide, I am pleased to introduce to you, and by the way, Slido is the way that we solicit questions from our audience members, as well as interesting polls. And so if you want to list where you're from on Slido, that's open right now. It's really a pleasure to introduce a colleague and a friend, Dr. John Kaltman. He's a medical doctor and the chief of the Heart Development and Structural Disease Branch at the National Heart, Lung, and Blood Institute. He is also an adjunct professor of pediatrics at Children's National Medical Center. Dr. Kaltman completed his pediatric cardiology and electrophysiology fellowship training at the Children's Hospital of Philadelphia. Here at NHLBI, Dr. Kaltman is responsible for developing new research initiatives in pediatric cardiology. He also provides leadership for the Biodata Catalyst program and the Pediatric Cardiac Genomics Consortium. His branch oversees the Pediatric Heart Network and Sudden Death Studies in Young Initiative. It's a pleasure to introduce him and for him to introduce the programs and data sharing, in particular the work of Biodata Catalyst and others at NHLBI. And so with that, Dr. Kaltman, I am passing the ball to you. And thank you again.
Thank you so much, Susan. It's a pleasure to be here today and participate in this session. Before I introduce our featured speaker, I thought I would just provide a very brief overview of some of the data sharing efforts that NHLBI engages in. Obviously, NHLBI is highly committed to sharing the various data and data resources that are funded through our institute. I'll mention just a few of the many efforts that we do, starting one with BioLink, which is NHLBI's biospecimen and data repository. BioLink has been in existence since 2009, and in terms of data has currently controlled access portfolio that includes over 55 observational studies, which includes over 780,000 participants and 176 trials, which includes over 460,000 participants. In 2020 alone, BioLink had over 375 data access requests. Many legacy studies as well as other studies have genomic data that have been deposited in dbGaP. And in particular, dbGaP currently houses 279 studies from NHLBI. And in 2020, had over 9,500 data access requests for those studies. And our newest platform for data sharing and reuse is Biodata Catalyst, and our speaker is going to talk quite a bit about that today. But I provided the link for the audience, so if you want to check out Biodata Catalyst, you can find it. Currently, Biodata Catalyst has over 3.42 petabytes of data, the majority of which at the moment is from our TOPMed study, which is our a study that's doing whole genome sequencing on various cohort studies that NHLBI has funded. Biodata Catalyst will also house NHLBI-funded COVID-related studies. We currently have one study, the ORCID trial, which is publicly available. And as soon as other studies are completed and published, we will get those into Biodata Catalyst as well. So next slide, please. So I'm pleased to introduce Elisa Manning. Dr. Manning is one of the principal investigators for the Biodata Catalyst Consortium, and we're thrilled to have her speak today. Dr. Manning received her undergraduate degree from Boston University, as well as a master's and PhD from Boston University in biostatistics. She did postdoctoral work at the Broad and Mass General Hospital, all in Boston. She currently is an assistant investigator in the clinical and translational epidemiology unit at the Mongan Institute and Massachusetts General Hospital, where she closely collaborates with the metabolism program and data science platform at the Broad Institute. She's also an instructor of medicine in the Department of Medicine at Harvard Medical School. Dr. Manning co-leads the NHLBI Top Med Studies Diabetes Working Group, as well as their analysis committee, where she tackles methodologic and implementation challenges in genetic association studies of whole genome sequence data in massive cohorts. So with that, I will turn it over to Dr. Manning, who we are thrilled to have today.
Thank you very much. I am going to share my screen and get started. All right, so I'm really pleased to have the opportunity to give this talk, which I have given the title of Opportunities for NIH Cloud Interoperability Approaches to Improve Outcomes of Pediatric Diseases. My outline for the talk today will encompass 5 different sections that are quite different, really highlighting the interdisciplinary work that I do. I'm going to introduce myself and where my research team sits in the sphere of cloud computing, talk a bit about Biodata Catalyst and the NIH Cloud Platform Interoperability Efforts, the NCPI, at which time I'll switch over to a use case, which will demonstrate an actual project we're trying to accomplish using interoperability features, and then wrap up with some important data governance lessons. So what is the researcher journey? What is my researcher journey? My research lab uses complex trait genetics to study the genetic causes of diseases like type 2 diabetes and cardiovascular disease. Early on, a couple of years ago, as part of the Top Med Diabetes Working Group, I led a cloud computing pilot on the FireCloud platform, and we were the first group to extend the functionality of FireCloud to run genome-wide association studies. I received my first R01 from the NHLBI to develop methods for large-scale gene-environment interaction studies, and I wrote in my proposal that I would use FAIR approaches to writing our software and distributing workflows for cloud-based analysis. Our method, GEM, which stands for Gene Environment Interaction for Millions, has been recently accepted and will be soon out in the Bioinformatics Journal. I also became involved in generating training material to enable researchers' cloud-based genetic analysis. Now, most recently, I've become a site PI for a team involved in developing the Biodata Catalyst ecosystem. I have been involved in bringing trainees and collaborators into Biodata Catalyst through the Biodata Catalyst Fellows Program and a recent small effort to onboard researchers via a pilot public cohort offering. Today, I will present a project funded by the ODSS, a Cloud IC Interoperability pilot project. So a problem that we're going to address is I am a researcher. I have a research question and an analysis plan. Some aspects of the analysis will always stay the same. I will obtain approval to use some data sets in my analysis through dbGaP. I will set up a place to do my analysis, which will follow the training and authorization policies of my institution. And as a principal investigator, I will be managing my research team to ensure the proper use of the data sets. So in the past, we would use dbGaP functionality to find the data sets. We would use the dbGaP website to download encrypted data files, then decrypt them, and then we would write the scripts and documentation on how we organized our files for analysis. We could perform our analysis on a local computer or a high-performance cluster made available by my institution. So some disadvantages of this approach is that there are many steps. There are really large files. There are technical processes that need to be learned by myself or my research team. And I am unable to work directly with approved collaborators on my analysis of these data sets. So now I can use a web portal in Biodata Catalyst to access and organize the files associated with my approved dbGaP project. I could export links to the large data files to an analysis workspace, which has tools, apps, and workflows that enable me to implement my analysis plan. Importantly, there is an automatic check for permission to use these data files at the point in time where I'm accessing these data files from my analysis workspace. So while I'm managing the use of the data by myself and my research team, these features enable me to get to my analysis faster and more easily, and I can safely and securely work together with approved collaborators in a workspace. So very quickly, in a couple slides, I'm going to talk a bit about Biodata Catalyst from a graphical perspective. So I am really privileged to be able to benefit from Biodata Catalyst as a user developing methods and making discoveries with genomics data using components of the Biodata Catalyst ecosystem, which is depicted here in the graphic. So my research has really accelerated towards our overall goal of helping patients. There we go. So here's another graphic depicting components of Biodata Catalyst ecosystem. The features we will see today are the data search and cohort formation tool powered by Gen3. I use workflows that are registered in DocStore, and I will show you how we're using cloud-based secure workspaces on the Terra platform. And along the way, I'm going to highlight other platforms that we've used in our pilot analysis with, from projects other than Biodata Catalyst. So as you can imagine, many teams are working together to further the goals of further the vision and the goals of Biodata Catalyst. Through collaborative development, we have tackled many of the platform interoperability challenges that all IT stacks are facing today. So now I'm going to give a very brief introduction to the NIH Cloud Platform Interoperability effort and focus on the working group hosting my CICI pilot project as a researcher use case. The NCPI was created as an outcome of the NIH workshop on cloud-based platforms interoperability held in October of 2019. The NCPI has a goal to establish and implement guidelines and technical standards to empower end-user analyses across participating platforms and facilitate the realization of a trans-NIH federated data ecosystem. In addition to Biodata Catalyst, there are three other participating platforms: the NHGRI's Genomic Analysis Visualization and Informatics Lab Space, or ANVIL, the NIH Common Fund's Gabrielle Miller Kids First Pediatric Research Program, or Kids First, and the National Cancer Institute's Cancer Research Data Commons. My pilot project will focus on ANVIL and Kids First in addition to Biodata Catalyst. So what's our motivation for wanting to access data across ICs and stacks? Well, there's a lot of data out there, and sometimes data is generated on different platforms, programs, and data sets that would be useful to combine in the efforts of answering our big research questions. So the Systems Operation Working Group is one of several working groups in NCPI. This particular working group spearheads technical improvements to the NCPI participating platforms that enable improved interoperability. So my pilot project is hosted by the Systems Interoperation Working Group as a researcher use case. So what is possible in terms of interoperability between NIH stacks last year and then moving towards this year? Well, as of early 2020, we have data portals where dbGaP project data is indexed, searchable, and available to researchers with approved dbGaP projects. Data can be exported to analysis workspaces associated with each portal. Right now, it is not so easy for researchers to find data across these projects and environments and use data sets in their analysis environment of choice. So I'm going to give a very short introduction to some of the standards-based interoperability features that will be discussed over the course of my pilot project presentation. I hope that we can all begin to become familiar with the terminology and try to get past a lot of the technical jargon that we encounter as we talk about these interoperability features. I will highlight that I don't build these systems. I place myself as a researcher using these systems who have the advantage of working with the individuals who do build these systems. So I perhaps get the items described and defined to me quite often, whereas others can sometimes get lost in a little bit of alphabet soup. So we're going to try to get past that so that we all know what it is that we're talking about when we talk about these interoperability features. So why are we concerned with standard-based interoperability features? Well, basically, we need to settle on one way of implementing a feature so that the teams can actually get started developing and implementing and making these features available to researchers such as myself. So it is a goal of the Global Alliance for Genomics and Health to adopt and distribute open standards for genomic data sharing. So the standards that are being discussed and strategized and implemented within G4GH are meant to facilitate the operation of various platforms as people try to work together. So two important standards for us one is a specification for the authorization to access data, and another is the construction of data repositories and access tools that can be used to find, organize, and export links to data. So later you will see where this authorization and passports and DRS, as we call it, fit into my researcher journey as I use the data available to me on these platforms. It is incredibly reassuring to see the availability of the NIH RAS login service, which implements the standards-based tools developed along with the Global Alliance for Genomics and Health. So now we're in several of my projects, we're able to use the NIH RAS login to authorize to create the authorized access to the dbGaP project datasets. Finally, I will introduce a technology called Portable Format for Bioinformatics, or PFB, developed by the Gen3 Data Commons. PFB is a mechanism for describing, packaging, and transporting complex data sets associated with the dbGaP project. PFB has been used successfully on many of the NCPI participating platforms. So what we have here is the model that a user would use to access, analyze, and report upon the data for which they have dbGaP approval to analyze. The user here would use a portal associated with various projects to index and to search through indexed data to find the data sets that they wish to use in their analysis. The search results are then handed off to a workspace associated with the project in which the researcher can create the scripts or use existing tools to do an analysis. And at the time when the data is accessed for the analysis, the DERS mechanism double checks and verifies approved access to that data set by the user requesting access. And so here's a sketch highlighting many of the accomplishments that were that we observed by the NCPI Systems Interoperation Working Group. Well, we have PFB is working, DERS is implemented, RAS is functional, and a number of the platforms are beginning to pull in and use all of these technologies, leading towards future interoperability efforts. Now, in 2021, we do want to work to finish the connections. So developers on these platforms are actively discussing and implementing and testing a mechanism for allowing researchers to access the data via the appropriate portal, but then hand off the data to an analysis workspace as appropriate and allowed, but of their choice. So when there are options available to a researcher, we have the technical ability to put our data in a particular workspace and do our analysis. So this is one of the pieces that we're still working on. And now I'm going to sketch through our use case, an actual pilot analysis to address a scientific question. Our scientific question is that we want to investigate genetic factors related to congenital heart defects in a study design that uses healthy controls from 2 NHLBI cohorts. We want to perform a pooled analysis of these data on, for example, the Anvil platform powered by Terra. So I'm describing here the steps that I would go through in the development of my analysis plan to decide how to answer my scientific question. I will highlight here that this pilot analysis doesn't go through all the steps that one would go through to verify the validity and robustness of your statistical analysis towards your conclusions, but I'm getting us to a point where we have the data organized in a way that enables such a rigorous analysis. So as we begin, I need to find the data that I'm going to use in my analysis. I have chosen to engage with the Pediatric Cardiac Genomics Consortium data sets, which is a study of participants with congenital heart defects. I have decided to leverage the Framingham Heart Study whole genome sequence data and the Jackson Heart Study whole genome sequence data to draw together a population of individuals who could act as controls in my statistical analysis. I have decided to use the Genotype Tissue Expression Project, the GTEx project, to characterize some of the findings that come out of my genetic association study. And now with each of these data sets, the question becomes, how do I access the data, where do I access the data, and how do I pull them together to do an analysis? So I was able to set up multiple workspaces and linkages to data in order to work with the data on the platform in which they're hosted. I have a Biodata Catalyst workspace. I have a Kids First workspace, and I have an Anvil workspace. I, of course, went through the process of proposing even this pilot project and gaining approval for this project through the appropriate mechanisms on dbGaP. And now, as I continue to examine data availability, I will note that the PCGC data has been sequenced by multiple programs. So subsets of the cohort exists as available for researchers, but in multiple programs. And these multiple programs have their own data portals, and the challenge then really becomes, well, how in the world can we bring this data together to do an analysis of the data that I have gained access and approval to do? I'll note that the PCGC data is available from the TopMed program and the Kids First program. Framingham Heart Study data and Jackson Heart Study data is available through TopMed, and the GTEx data is available through Anvil. So as we put together our study aims and decide how our analysis will proceed, I've, at this point in time, unable to put the data on the same platform for analysis. So I'll show you how we've obtained authorization to index, search, and export data to the appropriate cloud platform hosting that data. I've decided to use a great method called ProxyCat, the Proxy External Controls Association Test, which leverages summary statistics from individual level data to perform an association test of rare variants in particular regions of the genome. So I've chosen to analyze on a per gene basis the ratio of rare synonymous compared to non-synonymous variants between cases and controls. Then this would highlight those genes in the genome where there's a difference in the rate of genetic variation, and could highlight those genes that are important for increasing or even providing protective risk, protection against risk for congenital heart defects. So our approach will be to look for genes with a signal using whole genome sequence data sets, using a set of cases from the PCGC cohort and controls from Framingham Heart Study and Jackson Heart Study cohort. And when I start doing a deep dive into dbGaP and the data sets available, I'm able to find myself to the dbGaP projects that hold the data sets that I'm interested in. So these are the data sets that I included in my dbGaP project proposal, and which were approved for the use of this pilot project. And along the way, I became introduced to many of the individuals from the NCPI Interoper Working Group, and I was invited to use this project as a use case for helping to drive together these interoperability features. I will note that as I obtained access to the data sets, I had to pay close attention to the data use regulations and consent groups to which individuals in each study belonged. So while there's the potential for sets of data in these particular projects to not be allowed for the use I propose. So I've been paying very close attention to the various consent groups associated with each dbGaP project, so that I'm using data in the appropriate way from beginning to end. So what did we do? On each native cloud platform, we used the indexing features to search for the data sets we wish to use in this particular analysis. So I'm providing some graphics of how we use the platforms to search data. So we first on Biodata Catalyst, searching those data sets available on Biodata Catalyst. Now on the Kids First portal, I'm searching for the data sets associated with my approved dbGaP studies. And then on the Anvil, I'm able to access the t-text data set. So they may look different, and that's intentional, but they do work. This data exploration is a really great way of organizing the data prior from a conceptual view, and using these export features to deliver this organized data or links to the data in an organized fashion to a particular workspace. So just to briefly introduce my analysis plan, I did, and as I described, I did my preparation steps on separate ecosystems, but using the same authorization framework. So I was privileged to be able to automatically access the data on this platform because we have approval in dbGaP. So it's linked to that approval. And I didn't have to download any data to my personal computer at my office or onto the cluster at my institution to do any of the data preparation steps and the analysis. I'm looking at rare variants, and I'm going to focus on protein coding and exonic variants if I'm going to do a comparison of rates of variation in particular genes. The rest of these are just the details, so I'm going to skip ahead to start looking at some of the results. So on each one of the platforms, I am able to use the tools, workflows, and the features of that platform to create analysis summaries of the data on that platform. So a real advantage of these cloud-based platforms is that the tools that I'm used to using are available to me, sometimes in a slightly different way, but that's something we just get used to. But as we learn these platforms, but I'm able to generate the same sort of summaries using the same programming tools and the analysis tools that I'm used to using on my own computer or my own cluster. So if we take a quick look at the results from our analysis, I did have to get some summary statistics together in order to perform this association test. But because I was not able to pool the individual level data in a single workspace at this time, we created the gene level summaries on each platform, and then manually downloaded these gene level summaries, then uploaded them to a Biodata Catalyst workspace where we could use the tools on the Biodata Catalyst to perform the final steps of our association analysis. So as I said, this is just a pilot analysis showing a mechanism of doing the analysis we want to do, even though it's not exactly reaching the goals of our pilot project, which we hope to be able to accomplish in the next few months due to some of the future interoperability efforts that are currently underway. So where were we then? Where are we now? And where do we hope to be in the future? So where we are now, I've showed how we can use separate workspaces within individual ecosystems to do analyses. We've become it's easy to use each platform. The systems for documentation and user support are very robust, and being able to use the tools that we're used to using really facilitated the analyses on each one of the platforms. But in the future, we hope that once we obtain dbGaP access and are able to have a single sign-on that is uniform across all the different components of the data access, we will have one data workspace in which we can pull together links to data and do the analysis in one space without having to do any manual downloads or uploads. So obviously the goal is to just be able to access and analyze data from a single workspace, even though they're coming from multiple platforms. Well, there were some stumbles and roadblocks along the way. So we were really grateful that as we were trying to work this out, technology such as the PFB import to Terra was settled. And so, and DERS links, so links to data on various platforms have been solved. We note that different platforms may come up with different ways of defining, writing, and implementing workflows. For example, there's the CWL, which is a method for writing a workflow that is implemented on the Kavatica platform, which is used for Kids First, and a WIDL method of writing workflows, which is used on Terra. So we are keeping track of these challenges that we encounter along the way, and find that there are lessons to be learned as we start to use data that's accessed on different platforms. For example, we'll notice that there's a slight difference in one VCF file compared to another, and we'll wonder about some methods that were used for producing some of the data files that we're using. And sometimes it's a little bit of digging to answer our questions, but we did find that there was always a way to find the answers to our questions, and in the collaborative sense, people were willing to answer our questions as we brought them to everyone. Well, the last vignette that I'm going to talk about are some important data governance lessons that we've learned along the way, and some decisions that we've made to help move, to make progress in how different programs may share their data with researchers who may be involved in multiple programs. As you could see, I was trying to access data on both Kids First and Biodata Catalyst, and it made a lot of sense from a research perspective to want to do so. But it's not so easy navigating the data governance to make sure we're doing it correctly. So we're trying to bring together the Pediatric Cardiac Genetics Consortium study data for the first time in a cloud for researchers to use. I'll just review here that the data was available on multiple dbGaP project data sets. The PCGC data was available in multiple dbGaP data sets on 2 different platforms, the Kids First platform and the Biodata Catalyst platform. So, and the mechanisms through which we were going to combine the data wasn't clear. So the challenge here is to maintain each program's data governance of the data that they are making available to the research community. So the data is in a particular location. For example, Kids First data using the Kavatica analysis platform is on Amazon Web Services, whereas the TopMed data, which is on Biodata Catalyst, is on Google Cloud Platform services. So the challenge then is, how are we going to access data across services? Well, our requirement as individuals trying to solve this challenge for the benefit of the research community is to maintain the roles of each data steward. We have to maintain the data steward's control of authentication to use the data, and the data steward's management of the data through these index services, such as the DERS index, as I was describing previously. So our interim solution for data access for analysis of the data that we'd like to use in Data Catalyst workspaces are that we could take data and copy it to both an approved location on AWS and the Google Cloud Platform, all managed by the appropriate agencies and authorization, maintaining the correct authorization controls of this data. So each program will maintain their own index and authorization tool, and users will be able to see both data sets, the PCGC data sets on TopMed and from the Kids First portal interface. And our desire is to get to a point where the data can be searched and exported with using this PFB technology. DERS will be used to access the data But then at the end, at the compute workspace stage, a user could decide to be on Kids First or Biodata Catalyst. We'd be able to do the same analysis on either workspace, which we could do now, but it's just this painful process of some manual steps to get the data in the way or in the correct workspace, organized the way that we want to do. So we're not necessarily introducing a new concept. We're just enabling easier and more trackable and modern methods for getting the data into the workspaces that we'd like to where we would like to analyze them. So our goal is to enhance the interoperability capability within Biodata Catalyst. We really want to remove the need to have data mirrored in the Google Cloud Platform and the AWS clouds. And so in the future, we are moving towards a paradigm where we have a technology called signed URLs to all the data sets that will, as authorized researchers, access these data sets through the signed URLs. It's cloud agnostic. And so thanks to some work that's been done by many people to solve the problem of multiple clouds, right now we believe we can avoid data egress charges since the Kids First data is provided with free egress from AWS while providing access to all the data in Biodata Catalyst. So as we work through this process of our interim solution, we are going to be writing up a paper describing our novel approach and the interoperability features that have been implemented in order for us to achieve this interim solution so that we can describe how we met the challenges of data governance for the purposes of enabling the research of data sets that exist from different programs, but that should and could be put together for to enable progress in research. So with that, I'm going to wrap up my talk. Many great individuals for participating, facilitating, collaborating, and helping me co-lead many of these efforts. We really wouldn't be where I have found myself today without many of the efforts from developers, institutions, programs, and institutes that know that interoperability is needed. So I'm really looking forward to the discussion period that we're going to have after I stop my screen share, and I'll turn it over to John and anyone else who's going to be leading the discussion.
Thank you so much, Elisa. That was a fantastic talk. We really appreciate it. So we have, as you can see, some questions from the audience. I'll kind of read them out and hand it over to you for answers. So are data in NCPI member platforms duplicated in dbGaP?
Now, I think I would say no, because dbGaP has a very nice organization of data sets, and the structure through which a project cohort or probably a cohort or a project would submit data in dbGaP would create a self-organized structure of the data in dbGaP so that the data was not duplicated. Now, I can't promise you that that's the case for every data set in dbGaP because there are many, many valuable data sets in dbGaP. But from my experience, data is organized within dbGaP so as to create links with data generated by one program to data generated in another program through a parent project under which data associated with that project sits.
Great. Why did you choose to do your analysis in Biodata Catalyst? Was it a decision driven by how much data was there versus the other platforms?
Well, that's a great question. In many ways, I chose the Biodata Catalyst because I've had the most experience in Biodata Catalyst. As a researcher involved in the TopMed program, I saw how cloud computing systems and platforms were developed over the years, and as Biodata Catalyst has been adopted or developed by the NHLBI and has become a preferred location for hosting of TopMed data, it was natural for me to use Biodata Catalyst as a home base for this particular analysis. Although I will say the vision is for us to be able to use various platforms as appropriate and allowed according to our choice. So it's been really interesting getting to know the Kavatica platform, and in addition to Terra, which is developed by the Broad Institute. So this interoperability pilot has given me and the opportunity to learn more platforms so that I can understand the benefits of using one platform rather than another.
Okay. For variables that are common across data sets but collected by different platform IC, is there any data harmonization effort ongoing? And I'm going to add to that something that you and I have talked about, which is the promise of these platforms to enable reuse of harmonized data that perhaps a specific investigator is doing, and then enabling others to benefit from that.
Right. So I would say that there are some efforts going on to harmonize data, and the efforts are wide-ranging and specific to programs, but I would say are often meant to enable the analysis of that data within a program, like the NHLBI Biodata Catalyst, or and across programs. So there are many conversations going on around data harmonization. I do believe it's one of the biggest challenges that we have as researchers, because many of these data sets have been made available to us after being in existence for many years and been used for many valuable research problems. But now we're trying to bring them together to answer research problems that are novel research problems. And so harmonizing the concepts, the data sets, the variable names within the data sets is a major challenge. And it requires a lot of work for the researcher. I don't think that will ever go away. A researcher needs to know what their research question is. They need to do the work to decide the appropriate use of a data set to answer their question, and do due diligence to maintain the integrity of the data, which means careful examination of the data and appropriate statistical methods being applied along the way.
Great. In your use case, do you operate on process data, for instance, a VCF file, or raw data? Are process data consistent across studies?
Well, there's many different ways of approaching the use of genomic data for genetic studies of complex traits. Most recently, I use process data, which is a VCF file that has been developed and curated by a program or an initiative in order to enable analysis. I don't often handle raw data from sequencing platforms, but the need to harmonize the data will never go away. And for TopMed, for example, there was a massive effort to create VCF call sets, so genotype data call sets that were produced in such a way to enable the use of this data for many researchers and many purposes. So the problem of creating the data sets is being addressed by probably a program by program in a process that's led by the program, I guess I would say. But as people begin to combine data sets across the programs, careful thought has to be put into the appropriate use of the data and the correct application of methods. In fact, the statistical method that I proposed or introduced today was made in order to solve the problem of data sets being generated by different programs and a desire for a researcher to appropriately combine data sets, data from data sets that were generated at separate times and by potentially separate processes.
To what extent are the barriers to interoperability technical or policy, data use limitations, consent issues, et cetera?
Well, I would say that the barriers right now are technical, because the policy issues are issues that as the researcher community has been dealing with policy issues, data use limitations, and consent issues all along. In our analysis of genomic data for complex disease, we look at the data sets that we want to use to answer our questions, and we figure out how to use the data sets within the data use limitations and consent issues, never getting around any of the policy in using the data. So while there are always going to be steps to gain approval to do an analysis that you would wish to do, the policy will always be in place, but the interoperability features that are implemented should smooth access to the data set so that we can get to our, basically doing our analysis faster.
Okay. How easy is it to find data sets in the platforms such as Anvil or Biodata Catalyst?
So I would say that there are tools being built and made available on the Anvil and Biodata Catalyst portals, available to, if not to the researcher community right now, they would be made available soon, so that researchers can browse the available data sets. I know I've seen this in action, and I just don't know if it's available to the researcher community at this time. I would hope it would be, and I know that there is a vision so that individuals who want to do research with data sets can browse and examine the data sets that they wish to use prior to submitting a dbGaP application.
Okay. Following the question on cross-platform data harmonization, how well does search work across multiple platforms? Sort of same question.
I would say that that's a feature of interoperability feature that has yet to reach a developed state. I know there are some efforts in NCPI that begin to address cross-platform data harmonization, and so I look forward to learning more about those efforts and using them in the future. Right now, I think the search right now has been developed and is quite mature for a particular platform.
It looks like
I see the NCPI outreach group. I've learned about this search tool just this week. I actually just last week learned about the search tool, so I didn't feel comfortable throwing it out there, but I'm so glad that David Higgins has put this up in the chat. So great. Thank you so much.
Yeah. Can I bring my own analysis tools to the workspace?
Yes, I think given that your analysis tools are built within a paradigm allowed on a particular workspace. So one of the advantages of having multiple workspaces is that there's slightly different solutions may arise to perform analyses. So having your own analysis tool is possible, and I've done it in many different ways. I've written analysis tools that did quick ad hoc analysis. Once you get used to how to build an analysis tool, I can write a quick pipeline that uses a Linux-based tool fairly quickly. But we also have some more sophisticated tools that use Python programming or R programming or any tool that could exist in a Linux-based environment to do my analysis. So users can write simple analysis tools, they can write complicated analysis tools, and it's a vision of many of our programs to have these tools available and portable between systems.
Is it possible to access demographic, phenotypic, and clinical data without genomic data?
I would say yes, given that the data is available in a dbGaP project that has that is hosted by one of the programs of interest. So in a lot of ways, the data sets that you are using would sit in dbGaP, and these platforms are enabling access to that data. So and you could ignore some of the data sets and focus on some of the other data sets that are available and associated with a dbGaP project.
It sounds like your authorization was the same for each platform. Are there scenarios when you might have different authorization across platforms?
So let me clarify. I didn't mean to imply that the process was exactly the same. I think it's more appropriate at this point in time that the mechanism for obtaining access is the same. So I have an ERA Commons profile linked to a dbGaP project, which is an approved project with some data sets associated to it. And that mechanism for me as a researcher enabled me to gain access to, or be authorized to access the data for each platform. I know that there's a future vision using the NIH RAS login server, and that has begun to become implemented on the various platforms. So the technical solution may not be the same, but the researcher experience is the same.
All right, we'll end with one more question and then hand it back to Susan. If data in Biodata Catalyst is available in both AWS and GCP, why was there an issue in bringing the data from Kavatica AWS to Biodata Catalyst GCP?
Oh, I think that the simple question here is that there's just not that in our interim solution that we're proposing, we would wish to have the data hosted on both cloud platforms, but the issue is around data governance and where it's indexed and searched by a user so that each program can maintain the governance of their data. So it's not as simple as where the data sits. It's the policy surrounding the data from the program and platform perspective that will lead a researcher towards the appropriate route to seeing the data and then accessing the data.
Great. Thank you again so much, Dr. Manning. This was a fabulous talk and Q&A session. There are a lot of more questions, but unfortunately we're running out of time, so I'll hand it back to Susan.
Thank you so much, Dr. Manning and Dr. Kaltman. This was a great presentation. It will be made available on our website as soon as it's made 508 compliant. And I hope that you'll come back again to speak and also to listen to additional talks from across our NIH institutes and centers from PIs who are sharing data and reusing data. It's a pleasure to be the supporter of the NCPI Cloud Interoperability Platform and Program. It's just one of many activities my office supports to really catalyze trans-NIH capabilities in data science. And so with that, I want to thank you again. Come back in April, roughly same time frame and same time, noon, and we'll see you soon. Have a great March. Thanks, everyone.
Thank you very much. Have a nice night or day.
Happy Friday.